A very powerful another day, students. Um, this is a very positive thought I found today, that push yourself so hard, work hard so much, because no one else is going to do it for you. And more you will push yourself, success will definitely come to you. Remember that. If there will be little bit loose gaps in your hard work, then there can be then different results, right? So keep working hard. Don't think that what will happen after next few years. Just only concentrate on your hard work. If you will continuously push yourself, then definitely you will achieve success also. Okay, so with that positive energy, with that positive thought, let's talk about our third plant growth regulator, and this is cytokinins. As you know that we are talking about our chapter plant growth development, and from plant growth development we have taken plant growth regulators. So cytokinin is our third PGR. Cytokinins, when we say what are cytokinin, just look at its name. It means something they are going to do with the cell. And kinesis, you know that it means they are going to play a role in cell division, right? So here that it means they are the substances which play very helpful role in enhancing the process of cytokinesis. और cytokinesis क्या होती है? Division of cell cytoplasm during the cell division basically. So when cytoplasm divide करता है, so cytokinins, these molecules, these substances, they play role in division of cytoplasm. So there were few observations which were made by our scientist F. Skoog and Miller. They showed that cells of internodal area, suppose this is our stem portion, and from here leaves are coming out, so this is the area which is internodal area, right? So scientists observed that the cells in this particular area, and specifically the stem of tobacco plants, they show multiple or continuous cell division, means their cells, they show division very rapidly, and that is called proliferation. This observation was made by two scientists, Skoog and Miller. They observed that cells of internodal area of stem of tobacco plants, they showed proliferation. But why? Now they observe this division there, that these tobacco plant stem, they are showing the continuously division of cells. But when did they observe this division? Only when auxin that was added with the extract of vascular tissue, or coconut milk, or yeast extract, or DNA extract. So they said if auxin is added with any of these extract, then cells, they show proliferation or cell division. Or you can, in other terms, you can say if with auxin you add vascular tissue extract, or you add coconut milk, or you add yeast solution, or you add DNA extract, then continuously cells, they start showing division. But later on, Skoog and Miller, they identified, they discovered, they isolated these कि कुछ तो है यहाँ पे, कोई तो सब्सटेंस है जो सेल डिवीजन को इन्हांस कर रहा है. सो दे आइसोलेटेड दिस सब्सटेंस फ्रॉम herring sperm DNA. Herring is a fish, bacha. So they isolated this substance and they named it kinetin. Later on, scientists with their another experiments, they named it specifically cytokinins. They said because they playing a very important role in the cytoplasmic cell division, so they then changed the name to cytokinins. Still there, although now you know that these are the substances which play role in cell cytoplasm division, right? But chemically what it is, we know that cytokinases, they are purine derivatives. So chemically kinetin is 6-furfuryl aminopurine. It means it is a purine derivatives. It is derived from purine nitrogenous base and specifically adenine. So later on it was observed that cytokinin, they do not work alone. ऐसा नहीं है कि अगर स्टेम में इस एरिया में cytokinin release हो रहा है तो it will suddenly start showing the rapid cell division. No, cytokinases always work. They do not work alone. They always work with another phytohormones. Like I gave you one example, that is the auxin. So they do not work alone. They work with others. So if we want to see that how you can prove that they work with other, let's see their physiological effect. Now, suppose you have to grow your plant in the laboratory conditions. So here I have prepared a medium, right? Now I have taken the cell from the plant tissue. I have taken cells, right? Okay, I have just added cells in my medium. Now I have added auxin and cytokinin in the equal concentration, same amount, same concentration I have added them. And what will be our observation? That cell division will occur. Now if you added 4 cells, now there will be 8. If you added 8 cells, there will be 16 cells. So cell division will start occurring there, and this cell division will be without differentiation. ऐसा नहीं होगा कि इस सेल के अंदर से रूट्स निकलने लग जाएंगी. No, first there will be lot of number of cells. So this group of cell, that is called callus, and this is very commonly used in tissue culture only when cell division happens. So then we use equal concentration, equal amount of our auxin, A for auxin and C for cytokinin. Suppose now if I add, now after 1 week, if I add more auxin than the cytokinin, what will happen? Auxin initiate root. So now my roots, they will start developing, right? So roots will start developing there now. Now in this particular medium, I changed the medium and I added more concentration of auxin. So with the more concentration of auxin, root development will start there. After another few weeks, now I changed the medium and now I kept the concentration of cytokinin more. And I will observe that after root formation, now my shoot formation that will start, right? So as auxin, if it is more than cytokinin, that will result in root formation. If auxin is less than cytokinin or cytokinin is less, then it results in cytokinin is more, then it results in shoot formation. So as you can see that concentration of auxin and cytokinin, when you are changing their concentration, they are helping in differentiation. So cytokinin also play major role in differentiation process. So it plays a role in cell division as well as in your differentiation. So functions, which function you can write down? It promotes cell division. It promotes cell divide होगा तो cell बड़ा भी होगा. So it help in cell enlargement as well as cell differentiation. Now it inhibit apical dominance. It inhibit apical dominance. It means it promote lateral shoot growth as well as adventitious root formation. Adventitious roots means roots which develop from any tissue except रेडिकल. यूजली व्हेन सीड स्टार्ट जर्मिनेट, जो रूट्स डेवलप होती हैं वो किससे डेवलप होती है? रेडिकल से. बट एडवेंटिशियस रूट फॉर्मेशन इट डेवलप्स फ्रॉम एनी टिश्यू बट not from the radical. So cytokinin helps to promote the formation of adventitious roots. It also play role in the formation of leaf and their enlargement. It help in breaking seed dormancy. Dormant is the resting period when seed will not germinate. Even if it is sometime not germinate, even if the conditions are favorable. कई बार तो हम बोलते हैं कि dormancy is broken down when seeds come in touch with favorable condition. But sometimes even when the favorable conditions are there, sometimes still seed does not germinate because of many reasons. Sometimes metabolic rate is low, sometimes abiotic factors are not comfortable, so depending on the situation. So cytokinin it break seed dormancy by increasing metabolic activity in plants. So more easily the seed dormancy will be broken down, the plant will grow easily. So cytokinins break seed dormancy by increasing metabolic activity. Then another important function it plays, that is it delay senescence, means it keeps plant younger for a longer time. Now we are giving you two plants, the two leaves from the same plant. We are giving you two leaves from same plant, right? Both are equally green. Now one leaf we are keeping in the cytokinin medium and other we are keeping just as such. What will be the observations? Scientists, they took two leaves. One was dipped in cytokinin, other was kept as such. So the leaf which was kept in cytokinin medium, it was green for a very, very longer time. But the leaf which was kept as such, that changed into pale yellow color or died after some time. So it delays senescence and helps to keep plants younger for a longer time. That is the reason, because of this effect, cytokinins are also used to keep fruits and flowers which are you have plucked from the plant. To keep them fresh for a longer time, cytokinins can be used. And this effect of cytokinin is known as Richmond-Lang effect. The effect which helps to keep fruits and flowers fresh for a longer time, or you can say it helps to delay senescence. This is known as Richmond-Lang effect, after the name of the scientist who discovered this function of cytokinin. So this is all about cytokinin, its effect as well as its uses. Next plant growth regulator we are taking, that is ethylene.